Good afternoon, everyone. I am Mr. Rahul Patil, presently working as Assistant Professor at the Department of Zoology of Rayat Shikshan Sanstha's Balvant College, Vita. At the very outset, I would like to thank Dr. Humne sir, the founder of Botany for You, and the entire team of Botany for You for organizing this national workshop on CSIR, UGC, NET, JRF, and LS preparation in life sciences, and giving me an opportunity to become a part of this workshop. So dear friends, in this workshop today we are going to talk on very important topic from the cell communication and cell signaling, that is immunity, a defense system. And I'm trying to cover the maximum topics in this today's lecture. So we are aware that from last 10 to 11 months, the entire world is fighting against this COVID-19 pandemic. And recently we have good news that the Serum Institute of India and the Bharat Biotech, they have produced a vaccination, or we can say the vaccine against this coronavirus, like the Covaxins and the Covishield. And in future days, the vaccination program will be started. But in this pandemic situation, we have know that the importance of immunity, how that immune system is working against these foreign invaders. So everyone is trying to boost their immunity by taking the help of different types of the medicines, by taking the help of Ayurveda, by doing daily exercise, by doing yogas likewise. But we know that pathogens like bacterium, fungus, viruses, or other disease causing agents, the internal environment of an animal is a nearly ideal habitat. The animal body offers a ready source of nutrient, or we can say a protected setting and a means of a transport of new environment. From the perspective of we taking the example of coronavirus, or we can take the example of flu virus, we are the wonderful host. But from our vintage point, the infection from the coronavirus and the flu is not an ideal condition, or we can say it is not so ideal for us. But fortunately, adaptation has arisen over the course of evolution that protects the animal from these type of the different types of the pathogens. So due to the course of evolution, different types of the cells are evolved in the animal bodies, and they are trying to fight against these foreign invaders. But we know that every day we are exposed with the different types of the pathogens. Despite from the we can say the exposure of variety of pathogens, not all of us are infected. We can say fighting against the different types of the pathogens. So different peoples have the different types of the immune system, okay, their own immune system, and they are trying to protect themselves from the different foreign particles, or they are trying to fight against the different foreign particles. So in this today's topic, we are trying to cover the all aspects of immunity. We are trying to cover the different types of immunity. We are trying to cover the cells and organs of immune system, or we can say the how that particular immune system work. That all aspects we are trying to cover in this today's lecture. So let us start that. These are the contents of today's lecture. We are going to focus on the some introductory part of immunity. Then we'll switch on the cells and the organs of immune system. Then we'll talk about the different types of the immunity, like the innate immunity and the adaptive immunity. Then we talk about the antigen, that is the self antigen, or we can say the non-self antigen. What are the different requirements to become a antigen? Then we'll talk about the antibodies. Here we'll talk about the structure, different types of the antibodies and their functions. And lastly, we'll talk about how the different types of the cells are going to perform the immune system, or they are going to fight against these different types of the viruses, pathogens like viruses, bacteria, and the fungus. And lastly, we talk about the antibodies mediated immunity, that is the humoral immunity. And lastly, we talk about the complement activation, or we can say the proteins involved in the complement activation system. Okay, so let us start that. So everyone knows that the every country has their own force, like military, like navy, like air force, or we can say like a sleeper cell, and they togetherly trying to protect the countries from the, we can say, from the terrorist. Okay, so as like human bodies, or we can say the animal bodies, have the different types of the soldiers, and they are trying to protect the human bodies or the animal bodies from the foreign invaders like pathogens. Okay, so in this slide you can see that these are the some sort of the soldiers. They are togetherly working to fight against the different types of the pathogens. So dear friends, in this topic we will try to cover the different types of the structures, the and the functions of these type of the soldier. So let us start that. So before going to start the actual topic, we must know about what is immunity. So when we talk about the immunity is defined as a resistance to disease. The resistance power which is present in our bodies to fight against the different infectious diseases. The disease caused by the coronavirus, disease caused by a flu virus, or disease caused by a blood flu virus. Okay, so that is called as the immunity. But when we know that the cells, or we can say the tissue and molecules that mediate the resistance to infections, is called as the immune system. Or in other words, we can say that the system which is present in the animal bodies, we are trying to fight against these type of the infections, that is called as the immune system. Okay, and the coordinated reaction of these cells and molecules to infectious microbes is the immune response. So dear friends, suppose I'm infected with the bacteria, or I'm infected with the virus. My body gives response like the fever or like the inflammation, and these are the immune response. So we call as the immune response. So how that immune response are evolved, or we can say how which the different types of the cells are evolved in the immune response. So study of all these aspects is called as the immunology. Okay, so in other words, we can say that the immunology is the study of immune system and its response to invading the pathogen. It is the immunology. So dear friends, we are going to talk on the different types of the cells and organs of immune system. So before going to know about the cells and the organs of immune system, we must know about the process that called as the hematopoiesis. Because the hematopoiesis, or we can say this process, plays important role for the synthesis of, or we can say the production of different immune cells, and that is happen in the bone marrow. So we know that the bone marrow is present in the long bones of our bodies, and that bone marrow is having a special cell that is called as the hematopoietic stem cell. I just called as a stem cell because that cell has a property of self renewing and the differentiation. Okay, so that's why I just called as the stem cell. Okay, so what happened that under the influence of some growth factor like GATA1 and GATA2, that stem cell, that hematopoietic stem cell, is differentiated into the two different lineage, or we can say two different progenitor cells like the myeloid progenitor and like the lymphoid progenitor. Likewise, what happened that lymphoid progenitor, against under the influence of different growth factors, is differentiated into the different types of the immune cells. So let's see that first, how that lymphoid progenitor is going to convert into the different cells. So under the influence of some growth factors, the lymphoid progenitor cell is converted into the natural killer cell, that is the NK cell, and that cell plays important role in the innate immunity, or we can say this is the non-phagocytotic cells, and which gives the response that called as the ADCC, that is antibody dependent cell cytotoxicity. What is meant by ADCC, we will see in upcoming slides. Okay, so that particular NK cells is evolved, and we can say that plays a crucial role in the innate immunity. Okay, so meanwhile what happened that under the another influence of growth factor, that lymphoid progenitor cell is converted into the long cell, or the dendritic cell, that having a long dendrite, that's what is called as the dendritic cell, and that is called as the professional antigen presenting cell. Okay, so dendritic cells are called as the professional antigen presenting cells, or we can say they are called as the phagocytotic cells. They engulf the foreign materials, and they are trying to show some part of that foreign material so as to activate the different immune cells. So the lymphoid dendritic cells are called as the professional antigen presenting cells. Okay, they are going to present the antigen on their surface so as to activate the different immune cells to fight against this type of the antigen. Okay, then we'll talk about the under influence of some another growth factor. This lymphoid progenitor is converted into the T cell progenitor and the B cell progenitor cell. So what happened that the T progenitor cell, which is evolved in the bone marrow or which produced in the bone marrow, it is transported to the thymus for the maturation purpose. And under the influence of environment present in the thymus, that T progenitor cell is become mature and proliferate into the active cell that is called as the T helper cell and cytotoxic T lymphocyte, that is CTL cell. Okay, so this is about the T progenitor cell, how that particular T progenitor cell is become activated and what are the functions we will discuss in upcoming slides. So switch on the another progenitor cell that is called as the B cell progenitor cells. Actually B cell progenitor cell itself activated, or we can say it mature in the bone marrow and it converted into the B cells, that is called as the mature B cells. And finally that mature B cells under the influence of antigen is converted into the antibody producing cell, that is called as the plasma cell, and some of the B cells are converted into the memory cells that store the memory of that particular antigen. Okay, so this is about the lymphoid lineage. So switch on the another lineage that is called as the myeloid lineage. Okay, so what happened that the myeloid progenitor cell under the influence of some growth factors is converted into the leukocytes, that like the granulocytes and the non-granulocytes. When we talk about the granulocytes, like the monocytes progenitor cells, eosinophil progenitor cells and the basophil progenitor cells. Okay, finally that particular progenitor cells are differentiated into their destiny, like the neutrophils. The eosinophil progenitor cells is evolved into the eosinophil, and the basophil progenitor cell are converted into the basophil cell. So as we are aware that these neutrophil cells are called as the phagocytotic cells, that are going to phagocytose the foreign materials. The eosinophil cells plays important role in the helminths infection, like the ascaris, like platyhelminthes. So they are trying to kill that particular helminths. The basophil cells are plays important role in the hypersensitivity reaction, like type 1 hypersensitivity reaction. Now what happened that the basophil cell is also converted into the mast cell, or we can say that activate the mast cell, and that mast cell also plays a crucial role in the type 1 hypersensitivity reaction. Okay, so finally what happened that some of the granulocyte progenitor cell is converted into the monocytes, large nucleus containing cells, that is called as the monocyte, and that monocyte is converted into the macrophages cells or the true phagocytotic cells. These are the scavengers present in the blood. Okay, I just called as the scavenger because they are trying to we can say fight against this foreign material. They are searching the foreign materials and engulf that foreign materials and kill them. Okay, so that's why I called as the a scavenger. Then we'll talk about the another lineage that formed from the myeloid progenitor, that is called as the erythroid progenitor cells or the megakaryotes. When we talk about the megakaryotes, the small parts of the megakaryotes is formed into the platelets, and we know that these platelets played important role as become a barrier for the entry of we can say entry of foreign material from the infectious site. Okay, so infectious site is protected by this type of the platelet cells. Okay, then we'll talk about the erythroid progenitor cell. So that erythroid progenitor cell is transported, or we can say it converted into the erythrocyte cell. Okay, so what happened that these all cells come together and trying to protect the animal bodies against these different types of the pathogens. So we will see that one by one. Okay, so let's see that the importance of the B lymphocyte. Okay, suppose this is the B lymphocyte. You can see in this figure that B lymphocyte is having some sort of the receptor that is called as the BCR. So antibodies like IgD and IgM itself act as the receptor for the B lymphocyte. When that B lymphocyte exposed with the microbes or encounters the microbes, that B lymphocyte is activated, and finally it converted into the two different types of the cells, like the memory cells and the antibody producing cell that called as the plasma cells. Okay, now what happened that plasma cells is starting to produce the antibodies which is specific against this microbe. I'm just talking about the specific because this is not act against the different microbes. Okay, so what happened that antibody is now trying to neutralize the negative effect of that microbes, or also activate the phagocytotic cells, also activate the complement activation. So how that phagocytotic cell is activated and how the complementation is activated, we will see in upcoming lectures. So now we talk about the T lymphocyte. Okay, so you can see here the T lymphocyte which is having their own receptor that is called as the T cell receptor, that is TCR. Now what happened that the antigen presenting cells which is having the antigen and some small part of that antigen which is presented on the MSC molecule on their surface, that is called as the epitope. Okay, so this is the epitope of that particular antigen which is expressed on the MSC molecule. Okay, so what happened that now this epitope with the MSC molecule interact with the TCR, and now the TCR become activated. Okay, and this first signal is given to that particular T helper cell. Now what happened that due to the first signal, now the different transcription factor is activated, and that transcription factor acts on the different genes, so that genes are going to translate into the different types of the cytokines. Okay, so dear friends, the cytokines are the we can say the chemicals or the molecules which play important role in the immune system by the activation of different types of the immune cell. So some sort of the cytokines act on the itself to the T helper cell so as to proliferate into the different types of the T cells and also activate the B lymphocyte. Okay, so also this type of the cytokines activate the macrophages cells, also activate the neutrophil cells and carried out the different types of the functions. Okay, so then talk about the another cell that is called as the cytotoxic T lymphocyte, that is CTL cell. Okay, so CTL cell is also called as the CD8 marker cell, that is also having their own receptor that is called as the TCR receptor, that is T cell receptor. Okay, that TCR is associated with the particular CD8 positive co-receptor. Okay, so what happened here? If that infected cells expressing the microbial antigen on their surface, that is called as the MH2 class, and then this is about the epitope of that particular antigen. Now what happened, the that particular MSC class 2 and the TCR receptor interaction has been takes place, and now CTL is become activated, and what happened that CTL introduced some granazymes to that particular infected cell, so as that infected cell is now killed by the process of apoptosis, or we can say that is killed by the toxins that is secreted by the CTL cell. Okay, then we'll talk about the NK cell. It is a very important cell because as I told you that the natural killer cell plays important role in the innate immunity. Okay, so that we know that innate immunity is called as the inborn immunity. Okay, the immunity is present before the exposure with the different types of the pathogen, that is called as the innate immunity. So what happened that they under influence of the interferon gamma, the NK cell is become activated and they binds to the targeted cell and release the granazymes, or we can say release some toxic material, and that toxic material kills the infected, or we can say the targeted cell. Okay, so likewise these type of the different cells are working togetherly to fight against these different types of the pathogen. Okay, so here are how that particular cells are become activated. We will see in detail in upcoming slides, so I'm not going to focus the how that particular cells is become activated, how they are converted into the nerve cell to the effector cell. So let's see the organs involved in the immune system. So dear friends, here I am not going to discuss the structure and the function of the different organs of the immune system, because we have a less time to cover the maximum part from the immunity. So, dear friends, I'm giving here the quick review about the organs involved in the immune system. So, here we are going to talk about the first organ, that is called as the bone marrow, and we are aware that this bone marrow plays important role in the function of the production of different types of the immune cells by the process of hematopoiesis. So, as we know that the multiple bones like the femur, ilium, sternum, humor that support the process of hematopoiesis. Dear friends, when we take the cross section of these long bones, we can find here a large cavity. That cavity is called as the medullary cavity. And here we can see that the two different types of the niches, like the endosteal niche and the vascular niche, with having some sort of the different hematopoietic stem cells. So when we enlarging this part, we can find out here the blood vessels like the central sinus and the medullary arteries run through the center of the bone and form a network of capillaries that closely associated with the bones and the bone surface. So dear friend, the bone surface is called as the endosteum. So dear friends, the both cells that line the blood vessels and the cells that line the bone that generate a niche that support the hematopoietic stem cell for the self renewal and the differentiation activity. So as I told you that here are the two different types of the niche that called as the endosteal niche and the vascular niche. So when you talk about the endosteal niche, that niche provides a primitive environment for the development of the hematopoietic stem cells. So generally here the immature stem cells are there. When that immature cell become mature, they are transported to the another niche that is called as the vascular niche, and finally the fully mature, we can say the hematopoietic stem cells are transported from this vascular niche throughout the bodies through these central sinuses. So dear friends, here we are going to talk about the another system which is associated, or we can say which is play important role in the immune system, that is called as the lymphatic system. So here we can see that the network of the lymphatic system which is associated with the some important primary organs as well as the secondary organs. So the bone marrow and the thymus are the primary organs of the lymphatic system, or the we can say spleen and the lymph nodes are the secondary organs of the lymphatic system. So here we can see that the network of the lymphatic system which is associated with the some sort of the thoracic duct, right subclavian vein, right lymphatic duct, and the left subclavian veins. So that particular veins and the ducts are plays important role in the transportation of the we can say the lymphatic fluid throughout the body. So dear friends, we are talking about here the another important secondary organs of the lymphatic system, that is called as the lymph node. So in this figure we can see that that particular lymphatic system is associated with a large knot-like structure, and that large knot-like structure is called as the a lymph node, and that plays important role in the we can say in the immune system. So dear friends, it gives a micro environment for the development, maturation, and the activation of the different types of the we can say different types of the immune cells. So when we take the section of this particular lymph node, we can find the two different lymphatic vesicles. The one vesicle that is called as the afferent vesicles, or the another vesicle is called as the efferent vesicles. So these afferent vesicles are used for the entry of the antigens, or we can say the antigen presenting cells, B cells, and the T cells that enters throughout these efferent lymphatic vesicles. So when we take the cross section, we can find here the three different section of this lymphatic node. The outer one is called as the cortex, the middle one is called as the paracortex, and the innermost layer is called as the medulla region, where the mature B lymphocyte or the mature T lymphocytes are present, or we can say here the activated or activated T lymphocyte and the activated B lymphocytes are present here. So dear friends, when you talk about these cortex, here the cortex is the site for the presence of the B cell, and the paracortex is the site where the different types of the T cells are present. So dear friend, this is about the lymph node. Then we'll talk about the another secondary lymphoid organ that is called as the spleen. So when you talk about the spleen, spleen is the 5-inch long in the human adult and plays important role in the two different ways. Actually, the spleen is act as the graveyard for the dead blood cells, and it also plays important role in the immune system. Because here we can see here the two different types of the site here. That is, the one is called as the red pulp, and the another is called as the white pulp. So, dear friend, this red pulp is plays important role to destroy the dead red blood cells, and this white pulp is plays important role in the immune system. So, this is about. Okay, so this is the evolutionary distribution of lymphoid organs in the vertebrates. So I'm not going to discuss in detail about how that particular organs are evolved into different vertebrates. So let's switch on the another important topic, that is the different types of the immunity. So basically what happened that the immunity is classified into the broadly into the innate immunity and the adaptive immunity. So as we know that the innate immunity is also called as the inborn immunity, or the immunity present from the birth is called as the innate immunity, while the adaptive immunity is also called as the acquired immunity. So this innate immunity is further classified into the species immunity, racial immunity, and the individual innate immunity. So we're going to talk about the adaptive immunity, which is classified into the active adaptive immunity and the passive adaptive immunity. And further, they are also classified into the natural and the artificial adaptive immunity. So let's see one by one what are the different events are occurs in the innate immunity and the adaptive immunity. So first see about the what is innate immunity. So as we know that the innate immunity is also called as the natural or the native immunity, which consists of a cellular and the biochemical defense mechanism that are placed even before the infection and poised to respond rapidly to different infections. These mechanisms react only to microbes and not to the non-infectious substances, and they respond in a specially that the same way to repeated infection. So in other words, we can say that this innate immunity refers to a non-specific defense mechanism that come into play immediately or within an hour of an antigen appearance in the body. So these mechanisms include the physical barriers such as skin, chemical in the blood, or we can say the different cells involved in the immune system that are going to attack on the foreign cells, like the natural killer cells, like the dendritic cells, like the macrophage cells, like the eosinophil cell, like the neutrophil cells. They are the cells which plays important role in the innate immunity. So let's see that the different types of the innate immunity that is classified into the species immunity, racial immunity, and the individual immunity. So when we talk about the species immunity that acquire the resistance to the pathogen, that's shown by the all members of the particular species. We are taking the example of the human and birds. As we know that the anthracis infect the human being but not to the chicken. This is the best example of this species community that is evolved in the same member of the species. Then we talk about the racial community. Within a species, different races may show a different in susceptibility to the different infections. For example, the genetic resistance acquired in the African peoples against the Plasmodium falciparum, they do not cause the any infection after the plasmodium falciparum to the malaria. But when we talk about the peoples from the outside Africa, they are infected with the this type of the plasmodium falciparum. So this is the best example of the racial immunity. Then we'll talk about the individual immunity. The resistance to infection varies with the different individuals of the same race, or we can say in the same species. For example, the homozygous twins exhibit a similar resistance to the leprosy and the tuberculosis, As such, correction is not seen in the heterozygous twin, as we know that the basic difference between the homozygous and the heterozygous twin. So let's see that the different factors that influencing the innate immunity. These are the age, hormones, and the nutrition. So as we know that in fetus, the immune system is immature and it become started maturing in the adult stage. In old age, there is a gradual waning of immune system has been takes place. Polio infection and the chickenpox infection is highly severe in the children as compared to the adult one. So another factor is the hormones. The hypersecretion and the hyposecretion of the hormones enhance the susceptibility to the infection, such as the in diabetes mellitus condition, like the hypothyroidism in adult, in the adrenal dysfunction. The third factor that influencing the innate immunity is the nutrition. So as we know that the malnutrition predisposes to the bacterial infection. It gives a more chance to infect the bacteria to the a normal cell, or we can say the normal human. So this is about the factor that influence the innate immunity. Then we'll talk about the major functions of the vertebrate innate immunity, or we can say the innate immune system. So that recruit the innate immune system that recruit the immune cells to site of the infection through the production of chemical factors, including the specialized chemical mediator called as the cytokines. As we know that these cytokines are the chemicals which are going to activate the different types of the immune cells, so as that immune cells kill the particular pathogens. The second function is that the activation of the complement cascade to identify the bacteria, activate the different types of the cells, and promote the clearance of antibody complexes or the dead cells. As we know that this complement cascade plays important role in the innate immunity by the activating the different types of the immune cells, or also clearing the antibody complexes. The third function is that the identification and the removal of foreign substances which is present in organ, tissue, blood and lymph by specialized white blood cell. So as we know that the white blood cell like the neutrophil cell, eosinophil cell and the mast cell plays important role for the clearing the, or we can say the removal of the foreign substances which are present in the organs, tissues and the blood and the limbs. The another function is that the activation of the adaptive immune system. So as we know that during the innate immune system, what happened that the some cells that is called as the dendritic cells or the macrophages cells are engulfs the foreign materials, and that foreign materials is processed inside the macrophages cell or the dendritic cells, and a small part of that particular foreign material is expressed on the surface of that particular antigen presenting cells with the help of MSC1 and MSC2 proteins. Okay, so we can say the MSC1 and MSC2 receptor. So that type of the MSC1 and MSC2 receptor is recognized by the cells present in the adaptive immune system, and this interaction activates the different types of the cells like the B cells and the T lymphocytes, so as the secondary response has been generated, and that is called as the activation of adaptive immune system. The third function of this innate immunity is create a barrier that may be the physical barrier or may be the chemical barrier. So physical barrier is formed by the skin. As we know that the skin is a main barrier which protect from us from the entry of the foreign materials inside the bodies, or some chemicals barrier like the chemicals present, or we can say the proteins present inside the bloods. They are trying to protect our bodies from the entry of these different we can say different pathogens in the infected site. So this is about the different functions of the innate immune system. Then we'll talk about the another immune system, or the type of the immune system, that is the adaptive immune system. So when we compare this immune system with the innate immune system, we can see that this is inducible and develops slowly than innate response. So this is a specific kind of immunity and has a memory, therefore providing a long-term protection. As we know that there are some sort of cell like B cell and the T lymphocytes are activated under the influence of antigen presenting cells, and it proliferate into the plasma cells and the memory cells. That memory cells is now going to store the memory of the particular pathogens, and therefore they are going to provide a long-term protection against that particular bacteria. So for the activation of this adaptive immunity, we require to contact with the foreign particles. So when we become a contact with the foreign materials, the first innate immunity is activated, and this innate immunity activates the late response that is called as the adaptive immunity. So here when we talk about the adaptive immunity, is often subdivided into the two major type, that is depending on how the immunity was induced, and these are like the natural acquired immunity and the artificial acquired immunity. So natural acquired immunity is occurs through the contact with the disease causing agents when the contact was not a deliberate. So that means when we are exposed with the foreign materials or the disease causing agent, and when then we got the natural immunity, that is called as the natural acquired immunity. The second type is the artificial acquired immunity. That means this type of immunity is developed only through the deliberate action such as the vaccination. When we take a small part of the bacteria or the virus as the vaccine to fight against this that particular virus or the bacteria to develop a immunity, that is called as the artificial acquired immunity. For example, nowadays we are facing or we are fighting against this COVID-19, and we know that the vaccines against this COVID-19 is ready. And when we take the vaccines like the co-vaccines and the Covishield against this corona vaccines, at that time our body is artificially acquired against the that particular coronavirus. So then talk about the different types of the adaptive immunity, that already I told you that the basically adaptive immunity is classified into the active and the passive adaptive immunity. So let's see one by one. So first we see about the active immunity. So when we talk about the active immunity, which is further classified into the naturally acquired active immunity and the artificially acquired active immunity. So when we talk about the natural acquired active immunity, this occurs when the person is exposed to a live pathogen, and due to the exposure, what happened? The development of disease has been takes place, and that particular person become immune as a result of the primary immune response. So this is called as the a natural acquired active immunity. So the artificial acquired active immunity can be induced by a vaccine or by taking a substance that contain the antigen. So as we know that a vaccine is a small part of that particular antigen that going to stimulate a primary response against the antigen without causing any symptoms of the disease. Okay, so that is called as the artificial acquired active immunity. So there are different types of the vaccines that plays important role in the artificial acquired active immunity. These are the live vaccines, killed vaccines, and the bacterial products. So these are the some examples of the live vaccines like the BCG for the tuberculosis. As we know that the importance of the BCGs, BCG against the tuberculosis. Then the Sabin vaccines for the poliomyelitis. Then the another type of the vaccines are the killed vaccines like the vaccines used against this coronavirus like the Covishield and the co-vaccines. Some vaccines like the hepatitis B, some are the non-neural vaccines for use against the rabies viruses. The another vaccines like the bacterial product, like the tetanus toxoid vaccines or the diphtheria toxoid vaccines used against the diphtheria disease. So then we'll talk about the another type of the adaptive immunity, that is the passive immunity. So there are the different types of the passive immunity. The first one is the artificial acquired passive immunity and the natural acquired passive immunity. So first is that what is meant by natural acquired passive immunity? That immunity occurs during a pregnancy in which a certain antibodies are passed from the maternal side into the fetal bloodstream. Generally what happens, the antibody type that is called as the IgG is transferred from the maternal side into the fetal bloodstream, so as that particular blood acquires the unnatural acquired passive immunity. Then we'll talk about the artificial acquired passive immunity. which is a short-term immunization done by the injection of the antibodies such as a gamma globulin that are not produced by the recipient cell. Suppose the one human is infected with some diseases and some sort of the antibodies plays important role against that disease. So we are taking the antibodies against that disease, that is called as the artificially acquired passive immunity. Okay, so this is about the type of the different types of the adaptive immunity. So here we can see that the differentiation between the innate immunity and the adaptive immunity. So as we know that the primitive response or the immediate response against the pathogen which is happens in the innate immunity, and we know that the very less time required for the activation of the innate immunity, while the more time required for the activation of the adaptive immunity. As we know that the some cells of the immune system like the neutrophil cell, the macrophage cell, natural killer cell and the dendritic cell encounters the foreign materials and process it by the action of the releasing some sort of the toxins or the granozymes. And finally what happened that these dendritic cells are also plays important role, or we can say the macrophage cell also plays important role to activate the cells involved in the adaptive immunity. So there are some sort of the cytokines is also plays important role in the adaptive immunity, but they are released after the activation of the different transcription factor. Now that the different transcription factors bind to the cytokine genes, and finally the different cytokines will be released, and for that require a more time. So that's we can see that for the adaptive immunity we require a day, 2 days, or more than 2 to 3 days. Okay, so this is about we can say the small differentiation between the innate immunity and the adaptive immunity. Then we'll talk about this is the chart which differentiate the active immunity with the passive immunity. Then here we are going to talk about the humoral immunity, that is the antibody mediated immunity and the cell mediated immunity. So detail about this we will discuss in upcoming slides. So then we'll talk about the another important topic from the immunology, and that is the antigen. So as we know that the value of the immunology is given due to the presence of antigen. So antigen is act as the foreign material for the antibodies that provokes the immune response in the bodies. So we can say that an antigen is a molecule that induce an immune response in the bodies, that is called as the antigen. So the proteins, carbohydrates, lipids may act as the antigen for bodies, but they are the foreign to the human bodies or the animal bodies. So when we talk about the antigen, they are classified into the two different ways, like the exogenous antigen and the endogenous antigen. So exogenous antigen means that the antigen that have enters in the body or inside the body from outside environment by the process of inhalation, ingestion and the injection, that is called as the exogenous antigen. For example, as we know that the infection of the coronavirus is the exogenous one. Okay, then we'll talk about the endogenous antigen, the antigen that have been generated within previously normal cell as a result of normal cell metabolism or because of viral or intracellular bacterial infection. Suppose the bacteria is present inside our bodies, and that bacteria is now act as the antigen for our bodies, that is called as the endogenous antigen. So let's see that the some important terms used in the immunology. The first is called as the immunogen. So word immunogen is analogous to the antigen. So immunogen is a substance, or we can say it is a mixture of substance that has the ability to provoke the immune response if they are injected into the body of human or the bodies of the different types of the animals. So dear friends, the immunogen is able to initiate and the innate immune response firstly, and latter leading to the activation of adaptive immune response. So when we talk about the immunogenicity is the ability of foreign substances such as an antigen to provoke and the immune response in the body of human or the other animal is called as the immunogenicity. In other words, the immunogenicity is the ability to induce the humoral and the cell mediated immune response is known as the immunogenicity. Then we'll talk about the antigen specificity. It is a ability of the host cell to recognize an antigen specifically as a unique molecular entity and distinguish it from the other with accurate precision. So the antigen specificity is primarily due to the side chain conformation present on the antigen. Then we'll talk about the haptane. So haptane is a small molecule that can elicit an immune response only when they are attached to the large carrier such as the protein. Then we'll talk about the epitopes. Epitopes is the portion, or you can say the small portion of the antigen that recognized and bound by the antibody and also called as the antigenic determinant. Then we'll talk about the some determinants of the antigenicity. There are the list of the determinants like the size, chemical nature, and the foreignness. So when we talk about the size, those antigens have the more molecular weight that are the we can say that is the potent that they are act as the potent antigen. For example, the most effective immunogens tends to have a molecular mass is near about the 40,000 to the 60,000 Dalton. Okay, so for example, the tetanus toxoids, egg albumin, thyroglobulin are highly antigenic, while the insulin, which is near about 5,700 Dalton, are either non-antigenic or the weakly antigenic. We'll talk about the some chemical natures. Generally, the proteins and the polysaccharides are highly antigenic, while the lipids and the nucleic acids are less antigenic one. Then we'll talk about the foreignness. Only antigen that are foreign to the individual that induce the immune response. That antigen is act as the antigen. So foreignness is very important because the non-self antigen is started to provoke the immune response, not the self antigen. Then we'll talk about another very important topic from the immunity, that is the antibodies. So dear friends, these antibodies are also called as the immunoglobulin that are synthesized by the plasma cells that are proliferated from the activated B cells that constitute near about 25 to 30% of the total serum proteins. These are present in serums, tissue fluid and the mucosal surfaces. All antibodies are immunoglobulins, but all immunoglobulins may not be the antibodies, because these antibodies is made from the or we can see it is present from the immunoglobulin family, uh, and the we can see that here the basic structure of the antibodies. So antibodies are large y-shaped protein produced by the B cell that is used by the immune system to identify and neutralize the foreign object such as the bacteria, virus, or the different pathogens. The antibody recognizes a unique part of the foreign target that is called as the antigen, or we can say the epitope of that particular antigen. That are the composed of the four polypeptide chains, two identical light and the two identical heavy chains. That the heavy chains and the light chains are linked by a covalent bond that is called as the disulfide bond. The light chain similar in all immunoglobulins, while the light chains occurs in a two varieties like the kappas and the lambda chain. So light and the heavy chains are subdivided into the variable and the constant region, we will see in this figure. Each heavy and light chain contains the amino terminal in a variable region and the carboxy terminal in the constant region. So dear friends, the variable region which is formed by the light and the heavy chain, it is called as the antigen binding site. So let's see that we can clear the picture when we see this the figure. So we can see this is the heavy chain and these are the two short chains, and that two short chain again classified into the two different parts like the variable light chain and the constant light chain, while this particular heavy chain is also called classified into the one variable heavy chain and the three we can say three constant heavy chains. So when we talk about the antigen presentation that is formed by the variable region of these both light and the heavy chains, and which contain a CDR regions. So CDR is nothing but the cell complement determining region. Okay, so here are the different types of the when we talk about the one antibodies. The one antibody is having the six different types of the CDRs. So there are the one, two, three CDRs here. Here are the one, two, three CDRs. So that means so here there are the six different types of the CDRs are present. So whatever the CDRs are present in this one chain, that are also present in the another side of the antibody. So one can say that the only six different CDRs present in a single antibody structure. So when we talk about the structure of the antibodies, this region is called as the FC region, and this region is called as the Fab region, or where the antigen is attached to these antibodies, and the middle region is called as the hinge region. So this FC region is plays important role to attach with the different types of the antibodies, or we can say which is plays important role to attach the different types of the receptors present on the immune cells. So let's see that the different functions like the antibodies plays important role in the absonization, the max cytolysis, antibody dependent cellular cytotoxicity, neutralization of the exotoxins, neutralization of the viruses, preventing bacterial adherence to the host cell, agglutination of the microorganisms, immobilization of the bacteria and the protozoans. So we will discuss about what is meant by absonization, what are the MAC catalysis, ADCC in upcoming slides. So let's see that these are the different classes of the antibodies like the IgG, IgM, IgA, IgE, and IgD. Generally, it is pronounced as the GAMDA. That is G. After G, there is the antibody A, then M, then D, and then lastly E. Okay, so this classification depend upon the concentration of the antibodies present in the serum. So here we can see that the function of the different antibodies. So IgA plays a role in the localized defense mechanism in external secretion like the tears. IgD is involved in the recognition of the antigen by the B lymphocytes. IgE also involved in the allergic reaction. IgG is responsible for the complement fixation. IgM is also responsible for the complement fixation. So in the complement activation or the complement system, these both antibodies are plays crucial role. We will see in upcoming slide. So let's see that one by one what are the different structure of the antibody, antibody G, antibody A, antibody M, and antibody D. So let's start with the immunoglobulin G or the IgG. This is the most abundant class in the serum, which constitute near about 80% of total immunoglobulin present in the serum. They are present in the blood, plasma, and the tissue fluid also. It contains the less carbohydrate when we compare with this other immunoglobulin. It has a lifespan of 23 days, the longest of all of an immunoglobulin isotopes. It crosses the placenta and provides the natural immunity to the fetus and the neonates at the birth that I already told you that. So it acts against the bacteria and the viruses by the opsonization, neutralize the different types of the toxins, activate the complement by the classical pathways. Catabolism of IgG is unique in that it varies with the serum concentration. So there will be a question on the NATE set or the gate examination. This IgG activates which type of the complement, and that is the that answer of this question is the classical pathway of the complement. Then we can see the figure of the immunoglobulin, which shows the different chain like the heavy chains and the two light chains. Here is the hinge region that is bound by a covalent bond that is called as the disulfide bond. And as we know that there are the variable regions that act as the antigen binding site. Main important thing is that it is present in the monomeric form. Okay, so these are the some subclasses of the IgG, like the IgG1, IgG2, IgG3, and the IgG4. Among that, the IgG1, 3, and 4 cross the placenta and protect the fetus, while the IgG3 activates the complement, while the IgG1 and IG3 binds to the Fc receptor on the phagocytotic cells monocytes and the macrophages and mediate the opsonization of the different bacteria and the viruses. Then we'll talk about the another antibodies that is called as the immunoglobulin A, that is the IgA, which constitute near about the 10 to 15% of total immunoglobulin. This is the secretory antibodies which is present in the milk, saliva, tears, mucus of respiratory tract, digestive tract, and the genitourinary tract. In serum, it is exist in the monomeric form. Actually, it is in the dimeric form, but in serum, it exists as the monomeric form. An external secretion exists as a dimer called as the secretory hemoglobin. It has a J chain and the secretory fish that protect from the action of the proteolytic enzymes. And the half life of the IgA is near about 6 to 8 days. So how that particular secretion of the formation of the secretory IgA has been takes place, we can see here the dimeric IgA binds to the receptor on the surface of the epithelial cells that endocytosed and transported across the cell to the luminal surface. So after reaching the surface, the poly Ig receptor is cleaved, and the small portion of the receptor that remain attached to the IgG A dimer, and that is called as the secretory component. Now the secretory piece protects this IgA from the digestive enzymes and the denaturation by the bacterial proteases. So what happened that normally this dimeric form is attaches with the receptor present on the different types of the cell, that is called as the poly Ig receptor. And what happened, the endocytosis has been takes place, and finally a small part of this particular receptor is being attached with that particular antibodies, and finally that particular antibodies now act as the secretory antibody A. Then we'll talk about the function of the IgA. It provides the local immunity because it is a secretory immunity which is present mostly in the secretion like the tears, like the mucus present at the respiratory tract. Secretory IgA binds to the surface antigen of the microorganism and prevents its attachment and the invasion of the mucosal surfaces of the respiratory as well as the digestive tract by immune elimination. The secretory IgA provides the important lines of the defense against the Salmonella, Vibrio cholerae, and the influenza viruses and the poliovirus. So secretory IgA present in breast milk protect the newborn during the first month of the life. So one can say that the for the immunization or the immunization of the babies, the mother milk is very important because that mother milk is contain this IgA, which gives primary protection against the infection of the different bacteria, or we can say the different pathogens. IgA activates the complement by the alternative pathway. This is a very important question as far as the net set is concerned, because earlier we have discussed about the IgG plays important role in the activation of complement by activating the classical pathways, while the IgA plays important role in the activation of the complement by the activating the alternative pathway of the complement. The IgE also promote the phagocytosis and intracellular killing of the different microorganisms. Then we'll talk about the another antibodies, and the very large antibodies that is called as the immunoglobulin M, that is IgM. It accounts near about 5 to 10% of the total serum proteins. It is present in the 5 monomeric form, that's what is called as the pentameric unit. They are held together by a disulfide bond and the J chain. The molecular weight of IgM is near about 90,000 to the 1 lakh Dalton, and the half-life is about the 5 days. So most of the IgM is present intravascularly. Near about 80% of the IgMs are present intravascularly. They are present in a low concentration in the intracellular tissues fluids, because as we know that this fluid contain the abundant of the IgA antibodies. The IgM also IgM do not cross the placenta. The presence of IgM antibody in serum of newborn indicates the congenital infection. The earliest immunoglobulin to be synthesized by fetus is near about the a 20-week fetus. The first immunoglobulin to be produced in a primary response to antigen is the IgM. The IgM is relatively short-lived, hence its demonstration in the serum indicates the recent infection. The monomeric IgM appears on the surface of the unstimulated B lymphocytes and act as the receptor for the antigen presentation or the antigen binding. So IgM is plays important role as the receptor for the itself for the B lymphocytes. So this is the structure of the IgM. We can see that these are the 5 monomeric units and they are bind together by the J chain and the disulfide bond. Then we'll talk about the some functions of the IgM. It plays important role in the agglutination of the different bacteria because they have the 5 hands to the bacteria is killed by the different immune cells. The IgM is activate the complement by the classical pathway that I already told you that it causes the opsonization and the immune hemolysis. The IgM believed to be responsible for the protection against the blood invasion by the microorganism. Then we'll talk about the another important antibodies that is called as the immunoglobulin D. IgD makes up approximately 0.2% of the serum antibodies. It is very present in a small amount in the serum. IgG found on the surface of the B lymphocyte along with the monomeric IgM. So that's why it is act as the BCR, that is the B cell receptor. IgD may plays a role in the eliminating of the B lymphocyte generating self-reactive antibodies or autoantibodies. Then we'll talk about the immunoglobulin E, that is Ig. Ig makes near about not we can say point not 2% in the serum. Very a small amount is there. So most Ig is tightly bound to the basophil and the mast cell via its Fc receptor. So dear friends, the Ig is plays important role in the hypersensitivity reaction by activating the basophil cell and the mast cell via the interferon receptor present on the mast cells and the basophil cells. The Fc portion of the IgE is made against the parasitic worm and some arthropods can bind to the eosinophil cell that enabling the opsonization. So IgE also plays important role to kill the different pathogens or we can say the parasites worm like the helminths, like the platyhelminths, like the ascaris. The Fc portion of the IgE can bind to the mast cell and the basophil cell where it mediate a many allergic reaction. The cross-linking of the cell-bound IgE by antigen trigger the release of the vasodilators for an inflammatory responses. So this is about the different types of the antibodies present in the human blood and that are going to protect our bodies from the different types of the infections. So this figure can reveals that the antigen capturing and the processing of the antigen on the MSC class molecule 1 and the class 2 MSC molecule. So as we know that the endogenous antigens are expressed on the MSC class 1 while the exogenous pathways are expressed on the MSC class 2 molecules. So what is meant by endogenous and what is meant by exogenous pathway that we have discussed in our earlier slides. So let's switch on. There are some sort of the receptor present on the B cells and the T cells, and they are plays important role in the activation and the proliferation of the both lymphocyte. So firstly, we talk about the receptor present on the B cells. So as we know that the B cell has their own receptor that is called as the BCR that may be the antibodies like IgM or the antibody D. Okay, that act as the BCR. So with this BCR, there are some sort of the co-receptors are there that is called as the CD21 receptor like CD19 receptor or we can say that the CD81 receptor which is called as the serpentine receptor is also present with the B cell receptors. So dear friends, these CD21 receptors binds with the C3D that is the protein of the complement system which is attaches to the antigen and which co-stimulate this receptor of the BCR so as that B cell is become activated and proliferate into the different types of the lineage like the plasma cells and the memory cell. So this is about the T cell receptor. So this is the T cells and this is the antigen presenting cells that is going to present the antigen on the class 2 MSC molecule. So this is the TCR, that is called as the T cell receptor, that is associated with some proteins like delta, epsilon, gamma, that is called as the CD3 receptors, which is also act as the co-receptor. And another important thing is that the CD4 receptor, which plays important role to give the co-stimulation to the activation of these T lymphocyte. So okay, so let's see the selection process of T lymphocyte. It's very interesting things. As we know that in the bone marrow, the progenitor T lymphocyte are synthesized, and for the maturation, they are transported into the thymus, and where inside the thymus they are selected and trained to fight against the different pathogens, bacteria, viruses. So what happened that inside the thymus, that particular stem cells are divided into the different types of uh, the T lymphocytes, like the double negative T lymphocytes and the pre-T lymphocytes and the double positive T lymphocytes. So when we talk about the pre-T lymphocytes, which is having a beta chain receptor or the expression of the beta chain receptor on their cell surface. So now when we talk about the double positive, the complete expression of the TCR as well as the co-stimulator has been takes place, and that's why it is called as the double positive, that is the CD4 positive and the CD8 T cells or the immature T cells. Now these immature cells are exposed to the that particular antigen presenting cell present in the thymus. These antigen presenting cells phagocytose the self antigen and showcase into the MSC1 and the MSC2 proteins for the activation, or we can say the for the maturation of this double positive T helper cell. So what happened that some of the cells of the double positive cell when interact with the MSC2 molecules by a weak recognition, they are mature as the CD4 T cells, and that is called as the T helper cells, and that process is called as the positive selection. And some of the double positive cells react with the MSC1 molecules of the antigen presenting cells by the weak recognition that are matured into the CD8 cells, or also called as the cytotoxic T lymphocytes. This is also called as the positive reaction. But what happened that if there is no recognition has been takes place between the any class of the MSC molecules, then that particular double positive cells are triggered by the or killed by the process of apoptosis. So dear friends, what happened here in the next place that is very important, when the strong recognition has been takes place either with the MSC class 1 and the class 2 molecule, that double positive cells also killed by the process of apoptosis, because there will be a chance of the we can say the autoimmune response. So this is about the selection of the T cells by the process of positive selection and the negative selection. So dear friends, these positively selected cells are then transported into the lymphatic organs for the we can say for the activation purpose. So let's see the activation process of the T cells inside the lymphoid organ. As we know that for the activation of the T cells inside the lymphoid organs, we require the various stimulus. So that's why I'm just excluding this part and switching on or enlarging the part where the different types of the receptors and the ligands are plays important role in the activation of the T cell or the different types of the T cells. So this figure can shows that how the that particular receptors and the ligands are important for the activation of the T cells. Suppose only, as we know that particular naive T cells, as I called as the naive T cells, because it is activated cells, or we can say this is the mature cells, but it is not yet activated, or we can say it is not recognized actual antigen of the bacteria or the virus or the any pathogen. So that's what is called as the naive or the naive T cells. So what happened here? The antigen presenting cells like the dendritic cells, like the macrophages or B cells, they are engulf the microbes. They are killed by the enzymes present in the lysosomes, and a small part of that particular bacteria or virus, they are showcased into the MSC class 2 molecules, and that particular MSC class 2 molecules is interact with the TCR receptor. So this only interaction is not triggers the activation of the T cells, and finally
there is no response, or the we can say there is the functional inactivation. So in other words, we can say that suppose this is our self antigen. Okay, that time also, that signaling also not response, or we can say that signaling also not trigger the activation of the naive T cells. In counter current, we can see here the importance of the co-stimulator for the activation of the T cells. So we can see here the MSC molecules along with that particular antigen is interacted with the TCR. Meanwhile, what happened that the CD28 receptor is interacted with the B7 ligand, which is present on the antigen presenting cells, and due to this reaction, what happened that that particular antigen presenting cells is started to secrete the some cytokines like the interleukin 12. These interleukin 12s act on these we can say the naive T cells. Under the influence of this interleukin, what happened that that particular cells is started to synthesize the protein that called as the interleukin 2 receptor. Now this interleukin 2 receptor is expressed on the surface of T cell receptor, or we can say is expressed on the T cell surface. And finally, another important signal has been takes place, that is called as the CD40 ligand and the CD40 receptors. This signal is we can say it gives the signal to that particular new T cell to secrete the interleukin 2. So dear friends, now this interleukin 2 is act on itself. Suppose I am the T cells and I am secreting the interleukin 2. That interleukin 2 is act on the myself, so become I am activated. That is called as the autocrine signaling. So due to this autocrine signaling, that new naive T cells is become proliferated and differentiate into the different types of the T cells like the T helper or the different types of the cytotoxic T lymphocyte. So this is the role of the co-stimulation in the activation of the T cells. So as I discussed here, the three different signals are required for the activation in the naive T cell. That is the interaction between the TCR and the MSC molecule, interaction between the that B7 and the CD28 receptor, also interaction between the CD4 and the MSC molecule, as well as the importance of interleukin 12 and the interleukin 2. Okay, so these are the signaling which trigger the activation of the different types of the naive T cells. Okay, so then we'll talk about the function of the T helper cell. So as we know that now this T helper cell or the T cell is converted into the active T helper cell, and further this active T helper cell is classified into the two different types of the cell, that is T helper 1 and the T helper 2. But how they are classified or how they are differentiated, so let's see here. This is the macrophage cell or the dendritic cell. As we know that in our bodies there are three different types of the antigen presenting cells, like the B cell, dendritic cells, and the macrophage cells. They are called as the uh, what they are called as the antigen presenting cell. So this signaling that I already discussed, that the MSC molecule is interact with the TCR, which gives the a signal first. The we can say the B7 is interacted with the CD28 receptor, which gives the signal 2, and this signal 2 triggers the release of interleukin 12 by the this antigen presenting cells, which act on these particular T helper cells, so as that particular expression of the interleukin we can say interleukin receptor 2 is started to release on the surface of that particular T helper cells. And finally, due to the signal of that particular third signal, that particular naive T cells is started to secrete the interleukin 2, which is act autocrinely. And what happened that the fourth signal has been takes place, which release the two different types of the we can say two different types of the transcription factor, that is the T bit and the GATA3. Suppose that particular naive T cells are going to secrete the T bit, which influence the secretion of the interferon gamma. And finally, this new T helper cell is converted into the T helper 1 cell. If that particular naive cell is secreted, started to secrete the GATA3, under this GATA3, what happened? That particular naive T cell is going to secrete the different types of the cytokines like the interleukin 4, interleukin 5, and the interleukin 13. These type of the cells are going to convert into the another lineage of the T helper cell, and that is called as the T helper 2 cells. So in this figure we can see that the function of the T helper 1 and the T helper 2. So this part we have discussed in this figure, that is the differentiation between the T helper cell 1 and the T helper 2 cell. So what happened that now this T helper 1 cell is started to secrete the interferon gamma, and as you know that the macrophages cell have the receptor for this interferon gamma. So due to this interaction between the interferon gamma and the receptor present on the macrophages, their macrophages are become activated, and the enhancement of the microbial killing has been takes place. So another function of the T helper cell is that that particular T helper cell also secrete the interferon gamma, which plays important role in the complement binding, and the opsonization of the antibodies leads to the opsonization and the phagocytosis of the different types of the microbes. Then here we are going to talk about the functions of the T helper cell. These T helper 2 cells, as I told you, that secrete cytokines like the interleukin 4, interleukin 5, and interleukin 13. So what happened that these interleukin 4, which act on the beta cells or the B cells or the B lymphocyte, that triggers the isotype switching of the B cell, so as that particular B cells is going to produce the IgE. Now that IgE is interact with the helminths, or we can say it activates the mast cells or the basophil cells, leads to the inflammation or leads to the killing of the different types of the pathogens. Another function of this T helper cell 2 is the that particular cell release the interleukin 5, which activates the eosinophil, and that eosinophil release some sort of the cytotoxic molecules that directly kills the parasitic worm, and by that is the example of we're taking the example of helminth and the platyhelminths. Okay, so in that way the T helper cells are going to function or going to plays important role as a immune system. Then we'll talk about the activation of the macrophages by the T cells. So that already we have discussed that there are different types of the co-receptor which stimulate the activation of the macrophages. So dear friend, this is our T cells and this is the macrophage cell. As we know that the macrophage cell are called as the antigen presenting cells, or the we can say that are showcase the antigen on the MSC class 2 molecule. So what happened that when that particular MSC class 2 showcase the antigen, the interaction has been takes place between the TCR and this MSC, that is major histocompatibility complex 2, that trigger the first signals and lead to the activation of the genes present for the cytokines. So second signal is given by the interaction between the CD40 ligand, which is present on the CD40 cells, and the CD40 receptor present on the macrophages cell. It gives a second signal, and due to this second signal, what happened that this particular neo T cells is the T lymphocyte are started to secrete the interferon gamma, and now that interferon gamma is interact with the receptor present on the macrophages cell, then that triggers the phagocytosis by the activating the different types of the genes present inside the macrophages cell. And we can see here it also increase the reactive oxygen species inside the that particular macrophages, which kills the different types of the bacteria. This response also increase the expression of the MSC molecules and the co-stimular like the B7 molecules. uh, it also secretion leads to the secretion of the cytokines like the tumor necrosis factor, that is TNF, interleukin 1, the different types of the chemokines, and the interleukin 12 for the activation of the or the we can say the proliferation of the different types of the T helper cell. This is about the activation of the macrophages by T helper cell. Now let's see the function of the CTL cell. As we know that this CTL is called as the cytotoxic T lymphocyte. It is a ball of the toxin. So what happened that if that particular cell is infected with the bacteria or the pathogen, as we know that every cell act as the antigen presenting cell, that particular cell is phagocytose that particular antigen and showcase a small part of the that particular microbes as the antigen on the MSC class 1 molecule. I am talking about the MSC class molecule. For the activation of the CTL, we require the MSC class molecule, not MSC class 2 molecule. So what happened that After the interaction between the TCR and the MSC class 1 molecule, that particular CTL cell is become activated and started to release the cytotoxic or the granazymes by the process of exocytosis, and that particular granazymes directly pores that particular the membrane of that particular infected cells, and they kill of the targeted cells by releasing some sort of the granazymes. So these are the functions of the CTL cells. So CTL cell directly kills by the infected cells or the targeted cell by releasing granazymes, or we can say by releasing some sort of the different toxins. It also kills the cancerous cell. Recently studies also reveal that the CTL cell also plays important role in the we can say the killing of the different cancerous cell also. Then we'll talk about the switching the another important part of the immunity that is based on the humoral immunity, that is called as the selection of the B cell. So as like the T cell, the B cell selection has been takes place inside the bone marrow, not in the thymus, because as I told you that the selection of the T cells is takes place inside the thymus, while the selection of the B cell is takes place in the we can say in the bone marrow itself. So as we know that in the bone marrow, what happened that this selection process has been takes place through the various steps like the proliferation. Firstly, takes place the proliferation, then leads to the formation of the pre-BT antigen receptor expression. Then again proliferation finally leads to the antigen receptor expression, and finally positive and negative selection has been takes place. So what happened under some influence of the growth factor, these pro B or T cells is converted into the some cells that having the pre-BT cell express the one chain of the antigen, that is called as the pre-BT cells. Okay, so now what happened that these some cells which is express the one chain that are selected for the conal expansion, and those are failure to the express the different types of the antigen receptor, they are killed by the process of the apoptosis. Now these selected cells again we can say synthesize the remaining chain of the receptor, and finally they are called as the immature B cells that express the complete antigen receptor, and those are a failure to express the remaining part of the antigen receptor, they are killed by the process of antigen. Now those cells which completely synthesize the antigen receptor now exposed with the self antigen. Suppose there is the a weak interaction has been takes place between these self antigen, that cells are selected and mature for the reactions. Suppose there is no reaction has been takes place between the self antigen or we can say the self antigen and the antigen receptor present on the B cell, that are killed by the process of the apoptosis. If suppose there is a strong interaction has been takes place, that cells are also killed by the process of apoptosis, because this also generate the autoimmune response. So to avoid that, that particular cell are killed by the process of apoptosis. Then we'll talk about how that particular B cell is become activated. So as we know that the B cells are activated inside the we can say the inside the organs or the inside the organs of the lymphoid or lymphoid system. So what happened that the B cell presents antigen to the helper T cell. So as we know that the B cell itself act as the antigen presenting cells. They engulf the particular bacteria and killed by some sort of the enzyme present inside the particular B cells, and express a small part, or we can say a show, they showcase a small part into the MSC molecules. Okay, so what happened that due to the interaction between this MSC molecule expressed on the B cells and the TCR receptor present on the T helper cell triggers the first signal, lead to the interaction between the B7 and the CD28 receptor, and finally the third and very important we can say interaction is required for the activation of the particular B cell is the interaction between the CD40 ligand which is present on the B cell and the CD40 receptor present on the we can say on the T lymphocyte, sorry, on the B lymphocyte. So what happened that this interaction triggers the release of some sort of the cytokines. That particular cytokines is act on the B cells and leads to the proliferation and the differentiation of the B cells. Let's see the how that particular activated B cells again leads to the isotype switching. So, when the antigen-stimulated B lymphocytes may differentiate into the IgM antibody-secreting cells, or under the influencing of the different types of the CD40 ligand and the cytokines, some of the B cells may differentiate into the cell that produce the different types of the IgG heavy chain classes, like the IgM, IgG, IgE, and the IgA. And we know that the function of the different classes. So switching of the different types of the classes, that is called as the isotype switching, and that is influenced under the some cytokines like the interferon gamma, interleukin 2, then CD40 ligand, and that is the tumor growth factor beta. Okay, so this is about the isotype switching. Then we'll talk about the functions of the B cells. So as we are aware that the functions of the B cells are also called as the humoral immunity. So what happened that the activated B cell is started to secrete these antibodies. Now that antibodies is binds to the particular microbes and neutralize the effect of that particular microbe. But also it also act as the, or we can say it also plays important role in the opsonization and the phagocytosis of that particular microbes. It also called as the it plays important role in the antibody dependent cellular toxicity by activating the NK cell. It plays also role in the lysis of the different types of the microbes. It also plays an important role in the phagocytosis of the microbes opsonized with the complement fragment like the C3b, and also the antibodies also activate the complement activation. So how that particular antibodies activated the complement, we will see in the upcoming slide. So let's see that how the different how that particular antibodies are going to perform the different functions. So this figure shows the effect of the microbes without antibodies, and here this figure shows the effect of the antibodies on the we can say the microbes. So if there is no antibodies, what happened that microbes bind to the receptor present on the cells, and they are enters into that particular tissues and infect that tissues, leads to the killing of that particular tissues. Okay, but if there is the antibodies are there, that antibodies bind to that particular bacteria or the virus, and that neutralize the effect of that particular bacteria. In next slide we can see that that infected tissue is started to release some sort of the release of microbes from the infected cells and the infection of the adjacent cells. So what happened that if the bacterial load is increases in that particular infected cells, that infected cell is we can say a lysis has been takes place, and finally the thousands of these bacteria is released from this infected cell, and they are ready to infect with the adjacent cells or the nearby cells, and lead to the large infection of the different cells. If the antibodies are there and the cell lysis has been takes place, now that antibodies is binds to the different types of these bacteria and blocks the infection of the adjacent cell. The third function is the pathologic effect of toxins. As we know that the antibody blocks the binding of the toxins to the cellular receptor. So in that way, the B cells are plays important role, or we can say the antibodies are plays important role in the immunity or in the immunology. So then we'll talk about the another function that is called as the ADCC, that is antibody dependent cell type cytotoxicity. So as we know that the this is suppose these are the antibody coated cells, or we can say that the bacteria is having some surface antigen. That antigen is recognized by the important antibody that is called as the IgG, and that particularly cell is opsonized with the NK cells by the interaction between the Fc receptor and the Fc region of the IgG, leads to the activation of the NK cell, and that leads to the release of some granazymes or the cytotoxins, which directly act on these particular infected cells and lead to the killing of antibody coated cell. The another function is that the particular antibodies, that is the IgE, attached to the helminths, and now that and IgE interact with the Fc receptor present on the eosinophil, It activate the eosinophil, and also eosinophil release some sort of the granazymes and the cytotoxins, leads to the killing of that particular helminths. So this is called as the antibody dependent cellular cytotoxicity. That means cells are release some sort of the toxins lead to the killing of the microbes or the helminths. That's what is called as the cellular cytotoxicity. The complement activation. Here we are going to discuss about the how the complement system is activated. So as we know that the complement system having a group of 20 plasma protein, they plays important role in the innate immunity. As we know that the word complement means enhance or complements our immunity. So when we talk about the complement proteins, they are synthesized inside the liver and they are widely distributed among the body tissues and fluids. When we talk about the function of that particular uh, complement proteins, they comes into effect only when they become activated. So dear friends, the apart from these 20 different types of the complement proteins, the 11 proteins are mostly involved in the complement activation, and these are B1, C1 to C9, and the D complement protein, and they all are act as the enzyme precursors, catalyze a series of enzymatic reaction so as to produce the product of the complement system. So when we talk about the complement system, they are classified into the three different pathways, like the classical pathway, alternative pathway, and the lectin pathway. This classical pathway is operated when the antigen antibody reaction has been takes place, while the bacterial, we can say the alternative pathway is operated when the bacterial endotoxin interact with the complement protein. And finally, the lectin pathway is operated due to the mannose binding lectin protein, which is associated with the mannose or the lactose sugar present on the cell surface of that particular microbe. So let's see that one by one how that particular pathways are going to operate, and they are going to perform the different types of the function. So let's see with the classical pathway. As we know that the IgG antibodies binds to the cell surface antigen present on the microbe, and now what happened, the FC receptor of that particular IgG is interact with the a large protein of the complement that is called as the C1. So this interaction trigger the activation of the C1. Now what happened that this activated C1 is act on the C4 molecule. C4 molecule is the another protein of the complement system, and it cleaves into the a large and the small molecule. The small molecule is called as the C4A, and the a large molecule called as the C4B. So this large molecule again attaches with this C1 and convert into the C2 convertase. And finally, this convert the C2 complement protein into the C2A and the C2B. And finally, that C2A, that is small protein of the C2, is binds with the C2 convertase, which is C1, 4B, and 2A, converted into the another important protein, and that is called as the C3 convertase. Now, dear friends, this C3 convertase act on the C3 protein, and which cleaves into the a large subunit that is called as the C3b, and a small subunit that called as the C3a. Now what happened that this C3b is interact with the C3 convertase, that is a C2a and C4b, and forms as the C5 convertase. And finally this C5 convertase triggers the late step of the complement activation that we will discuss in next slide. So this is about the classical pathways. When we talk about the alternative pathways, the reaction are same like the classical pathways, but basic difference is that this is triggered by the bacterial endotoxins present on the surface of that particular bacteria, like the lipopolysaccharides, which is present at the outer membrane of the gram grammed bacteria. So when we talk about the alternative pathways, it is also similar to that of the classical pathway up to the formation of the C3 convertase. Now the C3 convertase also cleaves the C3 to the C3b and the C3a and forms the C5 convertase. And finally, they also trigger the late step of the complement activation. Then we'll talk about the lectin pathways. So as we know that this lectin pathway is triggered by one enzyme or that secreted by the liver that called as the mannose binding lactin. So dear friend, this mannose binding lactin protein is binds to the surface, or we can say the glucose is present on the surface of that particular microbe that called as the mannose. Generally, the mannose or the glucose are present in some bacteria like the candida or the salmonella. So that particular interaction triggers the activation of the lactic pathways, which is also similar to the alternative and the classical pathways. So finally, these same pathways also convert the 5 or activate the 5 convertase, and which is also operate the late step of the complement activation. So here we can see that what is exactly happen in the late phase of the complement activation. So here we can see that in the late phase of the complement activation, this C5 convertase is binds with, or we can say it cleaves the C5 to the C5b and the small unit that is the C5a. And now the whatever the small units are like the C3a, C2a, C3 or C4a or C5a plays important role in the different types of the immunity. So one can so here what happened that this a small unit is trigger the inflammation, while the a large subunit binds to the C5 convertase and triggers the binding of the C6, C7 and C8 and leads to the formation of the membrane attack complex, and that complex is activated and creates the pores on the bacterial cell wall and leads to the cell lysis by the osmosis process. So this is about the activation of the cell complement system. Now let's see that the different function of the complement activation. The first function is the opsonization and the phagocytosis. So as we know that this C3b binds with the endotoxin present on that particular bacteria and leads to the opsonization or activation of the different types of the phagocytotic cells lead to the phagocytosis of that particular microbes. Then another function is the complement mediated cytotoxicity or the cytolysis that is created by the activation of the membrane attack complex that we have already discussed. The third function is the stimulation of the inflammatory reaction that is caused by the activation of the different cells like the mast cells, neutrophil cells, and the basophil cells, which secrete the histamine and the serotonin leads to the inflammation. So as I already told you that the small proteins like C3a, C4b, C5a, which activate the different types of the immune cells like the neutrophil cell, mast cell, or the basophil cell, triggers to the release of some sort of the inflammatory products like the histamine and the serotonin. And finally, the destruction of that particular microbes has been takes place. So these are the some functions of the complement system. So dear friends, hope you understand the some important topics of the are the references that I used for the preparation of the today's lecture. uh, so thank you for your patience and all the best for your future examination. Thank you once again.